pleasure today to introduce two of the doctors who are going to be speaking with us. We know that as patients and as advocates, we are always looking ahead to what's next for us in the event that our current therapies, treatments don't work. And some of it's clinical trials, some of it's a secondary or a tertiary drug. But we're very happy to have two very prominent physicians, oncologists with us today. Dr. Suzanne George is here. She's supposed to be second on the agenda, but she's first on the agenda today. And we're really happy to have her. She's from Dana-Farber. Dana-Farber is a division, for those of you who are not familiar, of Harvard Medical School. So people who come from Dana-Farber are highly credentialed, very well educated, and they have much to share with us. So we're really happy to have you, Dr. George, and thank you for taking the day to be with us.
Thank you. Thanks for having me and inviting me to share some thoughts today. As you noted, this is part 2, but it could be part 1 because Bill, Cap, and I are just going to swap order. What I wanted to do today was to talk a little bit about some drugs that are in active clinical trials, specifically Blue-285 and DCC-2618. These are two compounds that are in the process of developing and soon to launch phase 3 international phase 3 randomized trials in advanced GIST. And because of that, I thought this would be a good opportunity to provide some background around the data that is supporting the development of the trials and just to increase awareness around what's coming. So, even though this isn't a CME or that I didn't have to show my disclosures, I think it's only appropriate to show my disclosures related to this presentation because our organization, our center, is participating in both studies from Blueprint and Decipher, in addition to other GIST studies from Flexicon and other companies. But I just want to highlight that we are participating in these two, and I am a scientific advisory board member for both companies. So I know that you guys all know, as you guys are well informed of GIST 101, so I won't go through that, but just to touch on a couple high points as to why we need more therapies. You all are very aware of that, but as you know, imatinib is an excellent inhibitor of KIT exon 11. It's also an inhibitor, but less potently so, of exon 9 mutant KIT mutations in GIST. It is relatively narrow spectrum. It's clearly revolutionized how we treat GIST in the first-line setting as well as in the adjuvant setting, but we know that resistance develops and is a challenge that we need to overcome. The data should tell us that the primary mechanism for resistance is the development of secondary KIT mutations that it is not effective against. Sunitinib, which is standard second-line therapy, is broader spectrum. It actually, de novo, hits KIT exon 11 very potently when it's the only mutation. It's a very potent inhibitor of exon 9 mutations in KIT, and it also hits exon 13 and 14 well, but it leaves out some of the other resistance mutations, particularly in the 17-18 region. But one of the challenges is that it's also a VEGF receptor inhibitor as well as a number of other kinases that it blocks, and because of this, we have additional challenges, including toxicities that are different than what we see with imatinib, as well as the emergence of additional resistance mutations that are not covered by this spectrum. Regorafenib, which is the standard third-line therapy in the countries that have access to it, is another broad spectrum, very potent kinase inhibitor. Again, de novo, it's a really potent exon 11 mutation inhibitor. It hits exon 17 and 18, sort of where Sutent doesn't hit in the secondary resistance patterns. Regorafenib does have activity, but it's extremely potent inhibitor of other non-KIT targets. Again, we know that secondary resistance is a challenge and toxicity can be a challenge with this compound. So when we think about where we are now, imatinib, sunitinib, regorafenib, we've clearly moved the field forward to where we were prior to the development and use of kinase inhibitors and GIST, but there's tremendous amounts more to be done, particularly in the advanced disease setting where resistance remains a major challenge. Duration of disease control has also been a challenge beyond first line. So as we know, with each line of therapy, if we look at the trials that have been published, the duration of benefit continues to get shorter. And that's really when you look at a population as a whole. For an individual, someone might be on regorafenib longer than they were on sunitinib, or might be on sunitinib longer than they were imatinib. That doesn't happen a lot, but it does happen on occasion. So for an individual, you can have benefit that extends with subsequent lines of therapy, but when you look at the studies as in aggregate, the duration of therapy tends to go down with subsequent lines. So we know that we need to do better, we need to do more. It's likely that through additional lines of therapy, we develop more heterogeneity of resistance mutations, so we're seeing more sites of KIT that are emerging with resistance and that are sort of popping through our barriers that we have with the current drugs. And I put this up, this next bullet point about the challenges of drug exposure. This is something that I think relates to what Norman was mentioning earlier in terms of adherence, and I also think that it really feeds in very much with toxicity profile. Sometimes the reason that people don't get adequate drug exposure is because the drug is giving them so much toxicity that it's hard for them to take it. Not just from an adherence perspective, but also from a dosing perspective. We may choose to lower the dose to try to optimize exposure, but how that's impacting disease control, we don't really have a very, it's really hard to quantitate. And I think that that's particularly true for second and third line therapies, sunitinib and regorafenib, where the toxicity profiles are much more, can be more challenging. And then this feeds into the last bullet point, really, which is just sort of highlighting the challenges of non-KIT-directed toxicities of these multi-targeted kinase inhibitors, where we need to be concerned, and we see frequently issues of hypertension, hands-on skin reaction, and diarrhea, which are often the reasons that we're lowering the dose of, say, sunitinib and regorafenib, which really has likely little to do with KIT, but more to do with these other kinases that the drugs are very potently inhibiting. So again, I want to highlight two ongoing trials of KIT inhibitors. One is Blue-285, and the second is BCC-2618. Both of these compounds have recent presentations at ASCO and at ESMO, and I'm actually just going to go through the data that's already been presented. I'm going to try to put it in context, and please feel free to ask. We can have questions at the end, and I want to make sure that it's clear so that we can understand the background as to why the new trials are being launched. And I'll talk about that a little bit too. So this is a presentation that Mike Heinrich put together. We all put it together, but he stood up at the podium and presented at ASCO of this year, summarizing clinical activity of Blue-285 in advanced GIST. Again, this is a visual that summarizes what we just talked about, that really imatinib has revolutionized the management of GIST, but we know that due to the heterogeneity, the various primary mutations, as well as the heterogeneity of resistance mutations in KIT involving the ATP binding pocket, as well as the activation loop, as well as primary resistant mutations in small subsets of GIST, such as the D842V mutation and PRGF receptor alpha, that there are clear gaps that we need to fill. And Blue-285 was developed as a very potent inhibitor of a number of mutations, particularly the PRGF receptor exon 18 mutation, and also this is structurally very similar to the exon 17 mutation in KIT. So when you find a drug that's really potent against the D842V PRGF receptor alpha mutation, it also tends to be active in this area as well, because structurally there's a lot of similarity here. Okay, and so because of that, and because we know that this area is clearly an area of challenge and later lines of therapy in patients with GIST, the Blue-285 was being studied specifically in the D842V mutant patients with PRGF and PRGF receptor alpha, but also in heavily pretreated KIT mutant patients. So this is an ongoing phase 1 study of this compound. It's written into two parts, part 1 and part 2. Part 1 is complete, part 2 is ongoing. And fundamentally, this was a standard what's called a 3+3 phase 1 study, dose escalation study, which I'm happy to explain, or I can skip if you guys want. Briefly, what it is it's just slow escalations where you enroll a small number of people, check safety, and then slowly escalate up to try to find the dose that is the safest, and at the same time is being monitored for efficacy, so benefit. It's been determined so far that the MTD, or the maximally tolerated dose, is 400 milligrams per day. And then once MTD is reached, the expansion, which is continuing to enroll, is enrolling into 2 primary cohorts. One is the D842V patients, and the second is patients who have unresectable or metastatic GIST, who have failed at least 2 lines of therapy, one of which has to be imatinib. So for the patients that were publicly presented at ASCO, as you can see, there were 72 patients treated, and I think what's really exciting from my perspective for both Lu-285 and DCC-2618 and ongoing studies that Bill will talk about, including from the Plexicon drug and others, is that these are drugs that from phase 1, from first entry into people, are being targeted, are being directed towards GIST patients. So we're able to start to see potential benefit in the phase 1 dose escalation. I mean, phase 1 drug development in oncology is changing so much in the area of personalized medicine anyways, but it's just important to note that these are drugs that are being developed and thought about from the get-go in this disease. So to have a phase 1 study with 72 patients of GIST is I think, a really remarkable effort. This has been an international study right from the get-go. As you can see, patients that have KIT mutations had a median number of 4 prior therapies, with some patients up to 11 prior therapies. The Peter Jeffrey receptor alpha group is much less, and that's because we know that there are less active agents in this patient population. Oops, I'm sorry. The majority of patients with KIT mutant disease had prior regorafenib, so you're talking 4th line and beyond, but that's not the case for the Peter Jeffrey receptor alpha group. So here is a CT scan of an example of a patient with a D842V mutant GIST, Peter Jeffrey receptor alpha, D842V mutant GIST. And again, just for background, this is a mutation that is highly resistant to imatinib. Sunitinib is likely not effective. Regorafenib has never really been commented upon one way or the other. But it's considered a refractory mutation. And this example shows a patient who, at baseline, had this is just one representative mesenteric lesion here, and then after 4 months, was essentially difficult to measure at all. It may be hidden in here. I find it very difficult to find. Here is another patient in the dose expansion with a large mass in the pelvis that after 2 months has reduced dramatically, very difficult to even see. So this kind of very dramatic response in this particular mutation, unique mutation of GIST has been remarkable, and something that at least since I've been doing this, I don't think any of us have ever seen this mutation respond this dramatically to any compound. And here is the aggregate results in terms of what's called a water pulp.
Doctor, wait, one question.
Yeah.
And what percentage of GIST patients have that kind of mutation?
Very small, single digits. So D842V, Peter Jeffrey receptor alpha mutant tumors, they occur in the stomach, so they're gastric. And this, I think, gets back to the point about mutation testing, because the gastric primary tumor that has a D842V mutation wouldn't be one that you would think about expecting response from imatinib, for example. But it's about 5% to 8% of gastric GISTs. Interestingly, the proportion of localized GISTs that have D842V is slightly higher than metastatic, so some have wondered if the metastatic potential may be slightly lower. But when you look at metastatic populations, it's probably in the 5% or less.
Okay.
But the challenge is that if one does have this, it's treated, we need to be thinking about it very differently with regard to treatment. But you're right, it's a very rare subtype. So in this is the summary of patients treated with this compound. in aggregate. And so the numbers, these various numbers here are the different dose levels that people were treated at. Again, this was a dose escalation study, so some patients received only 30 milligrams of the drug. Some people received 400 milligrams of the drug, even 600, but that was ultimately deemed not to be the best dose. But what you can see is that nearly everybody treated, specifically with the D842 mutation, primarily D842V, although some with other abnormalities in that region, had significant reduction of tumor burden. So this is shrinkage. And the only person that didn't shrink was somebody that had a pituitary receptor alpha mutation, but in a slightly different location. And that may be why this location was not sensitive. But everyone in the D842V region, at least treated so far, has had some shrinkage. Over half have developed what would traditionally be considered a partial response. This is a progression-free survival curve. So basically this curve just shows how long people are continuing on without progression on Blue-285 in this unique D842 population. And at this point, the average or the median progression-free survival actually hadn't even been reached yet because so many people were continuing on drug without progression. Overall, if you look at RESIST, and I'm just going to focus on RESIST for a second because that's what the FDA looks at, 60% of patients had an objective response, meaning shrinkage of more than 30%. The other 40% had stable disease as their best response, meaning lack of growth or shrinkage less than 30%. And then if you look at disease control rate, which is PR plus stable disease, all patients achieve that. And this tends to typically match up pretty well with the CHOI partial response. Now, if we shift gears a little and say, okay, it's clearly this is very dramatic, and for patients with this particular mutation, you know, this could be, this hopefully will be a great option for them in the future if and when the drug becomes approved. And even now could be an option for people if they're in a country where the drug is, where the current study is open. What about the more common types of GIST, which are patients with KIT mutant GIST? And this is an example of a patient treated with Blue-285, 300 milligrams per day, which is being treated in the dose escalation cohort. And as you can see, hepatic disease here at baseline, developing much more cystic or hypodense, which is typically a response pattern by imaging, and persisting through at least 7 cycles. This case, this person had 6 prior kinase inhibitors, primary mutation in exon 11, and secondary mutations in exon 13 and 18, which is not at all an uncommon pattern of secondary resistance. So this is suggesting that there is activity perhaps in the KIT mutant population as well. Another example of a case, 5 prior kinase inhibitors, primary exon 11 mutation, unclear exactly what the secondary mutations are. Small nodule here as an example that's reduced in size here. And they call this a partial response by RESIST as well as by CHOI. Now this slide summarizes as it says, it's a dose-dependent tumor reduction across multiple genotypes, basically meaning that, as I mentioned earlier, this is a dose escalation study, so some patients are treated at lower doses, some patients are treated at higher doses. The thought is that with higher doses and high exposure, you'll get more target inhibition and perhaps see more activity. And in the KIT population, if you arbitrarily make this designation between lower dose and higher dose, you can see a greater proportion of patients who seem to be having tumor reduction than here, which was our, the patients at the quite lower dose. I think it's most notable when you look above 100 milligrams actually here in the KIT mutant population. So there seems to be some dose dependency here to some extent. And then again, if we look at a similar curve to what we did with the D842V patients, if we look in the KIT population, you can see the median progression-free survival. So again, the median time that people are continuing on drug without the tumor growing significantly if you look at the doses of 300 milligrams or higher, was over 9 months. Now, we have to remember, these are small studies, this is a small number of patients. How this could potentially sort out in a bigger study will be the question of the phase 3 trial, but this is very promising for a late-line therapy. Particularly when we think about imatinib retreatment, which was done in a Korean study where patients were randomized to either best supportive care or imatinib after failure of imatinib and sunitinib. Median time to progression in that study was about 2 months, just under 2 months even. And so the suggestion of a 9-month median PFS, again, numbers are small, is very encouraging in this patient population. And we still have 2 patients out of a small number, but 8% of patients with with partial response. We are seeing some tumor shrinkage as well. Oops, I'm sorry. So I also want to highlight the adverse event profile with BLU-285. Again, small number of patients, although 72 really isn't that small, but relatively small number. And what we're seeing is nausea, fatigue, some vomiting, swelling, periorbital edema, similar to what's seen with imatinib, diarrhea, some peripheral edema, decreased appetite. But when you start to look at the severity, what you can see is that most patients are reporting only grade 1 and grade 2, so more moderate side effects across this spectrum. I think that as we continue to have more experience with the drug, we know this from every drug that's been developed, is the more experience you have with the drug, the more the toxicity and the adverse event profile matures, the more you really understand stability or lack of stability around these numbers in terms of what's more common, what's less common. But at least on first blush, in this first study, the toxicities have been manageable and they have been they've been manageable. And of note, we're not seeing the toxicities that become problematic with regorafenib and sunitinib that we discussed, like hypertension, hand-foot skin reaction, that can become a real challenge in some of our other therapies. So the conclusions as were presented at ASCO were, BLU-285 was well tolerated on a daily schedule, it's just a once-a-day dose, up to the maximally tolerated dose of 400 milligrams per day. I did not show this data, but looking at the PK data, so with the pharmacokinetic data from the drug, 300 and 400 milligrams a day actually provide pretty similar exposure. So it's not clear how much extra exposure you're getting once you go above 300. It seems that there's very strong activity in the D842V Peter-Jeff receptor alpha mutation that we were discussing earlier, with an overall response rate of 60%. Which is dramatic and really has never been reported in this disease type before. And currently, Blueprint Medicines, who's developing this drug, is looking for expedited paths for approval of the compound, particularly in this population. But as of now, the study does remain open for continued enrollment in the expansion arms. It's also important to note that BLU-285 does seem to be demonstrating important anti-tumor activity as well as in heavily pretreated KIT mutant GIST patients. And I think that's also equally important because this is a clear area of unmet need. And because of this latter point, there is encouraging data and there's planning underway and ongoing for a phase 3 randomized study that will be international, BLU-285 versus regorafenib in third-line GIST. So the details of that are not yet flushed out, but this is the anticipated next development strategy for this drug. So now I'm going to switch gears and focus on DCC 2618. As I mentioned earlier, I think these are the 2 drugs that are in the process of planning for their phase 3 trials to hopefully study more definitively in the advanced GIST population. And these are the 2 drugs that in clinic I get asked the most about. So again, I wanted to share that data with you. This is data that was presented by Philip Janku, who's actually a phase 1 doctor from MD Anderson. And this was just presented in early September at ESMO as an oral presentation. So the background is very similar to what we discussed with BLU-285, as well as in the introduction. What I do want to comment on is really that DCC 2618 is what's called a switch pocket inhibitor. So it again targets KIT, it just targets it in a slightly different structural conformation than some of the other compounds that we've had so far. Their preclinical program suggests activity across multiple KIT subtypes, multiple KIT mutations, at least in the lab. And this, similar to the Blue 285 study, is a first-in-human dose escalation study. The dose escalation portion of this study is also complete. Expansion is ongoing. This study right now is only open in the US, but there are plans to expand as well. The dose has been determined for expansion. It's 150 milligrams once per day. And they'll continue to do extensive monitoring to make sure that that's the optimal dose for study into the next phase of the drug development. So this is pretty standard, similar to other studies. Patients are taking the drug every day. There were scans performed every 2 cycles. This study had extensive next-generation sequencing of plasma DNA, which I know we're going to hear more about shortly, but I'll just give a little hint of how they included it into this study. And that was looking for KIT and PDGF receptor as well as other molecular alterations in the blood of people while they're on the study. But they also paired this with real-time tumor biopsies at the beginning of the study. And then again, patients had to have at least, in this study, it had to be at least 1 prior kinase inhibitor, which was imatinib, but they could have up to as many as they had had. And for this study, and this study was open to most solid tumors, but with a real focus was on just patients, and that's where the bulk of the approval has come from. This is a safety profile of the drug. Again, similar, the side effects in clinic feel a little different between the 2 drugs, but similar in that the majority of toxicities observed were grade 1 and 2, which is generally mild. They only had a couple of grade 3 toxicities that were drug-related, both of which were elevations in the lipase, but not clinically significant. So lipase is an enzyme that's made by the pancreas, and when it goes up, sometimes that can be a sign of pancreatitis or pancreatic injury, but it can also be somewhat of an unclear if it's clinically relevant. So in a number of kinase inhibitors in earlier drug development, we've seen lipases go up and come down, but it's pretty uncommon to see true clinical pancreatitis. So this is more of a laboratory finding. But again, this, interestingly, in contrast to Blue 285, this patient with DCC 2618, there have been some hand-foot skin reaction, and it's probably because there is a very low level of inhibition on VEGF, which may perhaps be contributing to this, but still, in my mind, the mechanism of action of this is unclear. So in this study, 57 patients with GIST have been enrolled. 62 years old is the median age. Baseline mutations are what would be expected for a refractory GIST population, the majority primary exon 11, some patients with exon 9, actually probably overrepresented exon 9, exon 17 primary, and D842V in a small number of patients. Again, heavily pretreated with a median number of priors of just over 3, with up to 7, all patients receiving prior imatinib, 3/4 of the patients receiving prior regorafenib, and the majority of patients received greater than 100 milligrams a day total dose, with the recommended dose at 150. So this is what's called a swimmer's plot. This is a duration of treatment, which is how they've opted to show this. What I would comment on is everybody in blue, each one of these lines represents an individual patient, an individual person that's on study. The yellow lines are people that have come off study for various reasons, most commonly due to disease progression. The blue lines are people that remain on study. So, we don't know exactly how long these folks' lines will be because they're still on study or when they'll turn to yellow. But clearly there's a proportion of patients who have been on the drug for approaching or over a year. This is the progression-free survival curve. If you look at all of the patients treated at over 100 milligrams per day, the median progression-free survival has not yet been reached. It's, will come in right around here. Hard to know where that is exactly, but it's right. So we don't have a number yet, but it will hit it at some point. And again, there appears, similar to Blue 285, there appears to be some degree of dose responsiveness, but that's completely expected in a phase 1 trial like this. And over 100 milligrams daily is thought to be the target dose. This is a similar waterfall plot to what we looked at before. So patients identified with red bars are over 100 milligrams per day, and blue bars is less than 100 milligrams per day. And what you can see is that, there's a proportion of patients that this drug isn't the right fit, similar to Blue 285. Sometimes the drugs, we still need to do better, for sure. But there are significant proportion of patients who have had disease stability or some reduction, with a number of patients actually hitting the threshold of a partial response, meaning greater than 30% of the tumor reduction. Greater than 30% reduction in tumor. Now this slide is, I'd say it's intriguing perhaps. I just want to take you through it a little bit, and again, we'll talk about DNA a little bit later today, but this slide basically summarizes, let me back up a second. The cfDNA is cell-free DNA, so it's basically measuring DNA in the blood, particularly the DNA that comes from the tumor or that we think is coming from the tumor. And we have a whole other talk on that, so I'm not going to get too much into it, except to say that within the context of this study, they had serial measurements of circulating free DNA in the plasma, and they wanted to see, what the company did was they said, let's just see how much certain types of DNA that might be reflective of certain types of mutations goes down over time. And what they saw, what they reported out, is that they seem to be seeing reductions in cell-free DNA reflective of a number of different mutations in KIT that we know are present in advanced GIST. So this could imply that the drug seems to be doing something to the DNA, the shedding of the DNA or what's present in the blood. The challenge here, I think, which is something I think we all need to take with a little bit of caution, is this isn't yet superimposed or correlated with disease growth or disease shrinkage or disease stability over time. I hope we'll be able to have that data at future meetings, but right now this is sort of just intriguing that it seems to be interacting with the DNA in some way and it seems to be going down. This, I think, is another interesting set of data that's coming from this study that will need to be matured over time. But this makes a correlation between the mutations in KIT that were found in the tumor at the time of the biopsy, at the initial initiation of the treatment, as well as the mutations that were found in the plasma at the time of initiation of treatment. And again, this is an area that will need to be flushed out and matured over time, but it does suggest that in many cases, what you can find in the tumor, you can find in the plasma. So this is a patient with an abnormality in KIT exon 9, and that was found in the plasma in KIT exon 9. But also that there may be, that there's likely heterogeneity within the GIST itself, as we all know from previous studies, that not all tumors evolve in the same way. And so a biopsy from a single lesion might reveal a single mutation, the primary driver mutation, in this case, let's say exon 9. But in the plasma, you can pick up a broader range of mutations. In this patient, it's actually an exon 9 plus an exon 11, which is pretty rare but can happen, an exon 11, 17 and 18. So, it's suggesting that perhaps the plasma might be able to pick up a broader set of mutations than a single tumor biopsy can. And I think that that's consistent with what we know about heterogeneity and knowing that not all tumors behave the same within a person. The plasma might be able to give us a better view. So this study remains ongoing. I think, again, similar to Blue 285, it's showing promise in the KIT mutant population, heavily pretreated. There are 3 cohorts that remain enrolling. Patients who are true second and third line patients, so people who have failed only imatinib and people that have failed only sunitinib. Patients who are true fourth line, so just beyond, it doesn't have to be regorafenib, but have had 3 lines, and then this would be the 4th line, and then 5th line patients. The study is open in multiple sites in the US, and Toronto is open as well. And they will plan to open some European sites starting in November, although that site list, to my understanding, isn't completely final yet. The fundamental objective is to further define the safety and the tolerability of DCC 2016 at the 150 milligram dose, and to evaluate its anti-tumor activity in GIST across these cohorts. Very similar to what the expansion phase of Blue 285 is doing. And as these two drugs are sort of running in parallel to study this patient population. And in conclusion, the conclusions that were made at ESMO, again, we've sort of gone through this, but the drug seems well tolerated. We're seeing some disease control, which I think is very encouraging, with disease control up to, at 12 weeks, at 3 months it's 76% at 3 months, but 57% lack of progression at 6 months, which is very promising in a heavily pretreated population. Again, these are early studies; they have to be confirmed in more patients. And we are seeing activity across the board in a wide number of KIT mutant patients. And then they're ongoing, as I mentioned before, the Blue 285, their planned phase 3 will be head-to-head against regorafenib, third line, international study. For DCC 2618, they have 2 planned phase 3 trials. Actually, the first one will be placebo-controlled in patients who have received at least 3 priors. So in the space in which there is not an approved therapy right now is where the next study for DCC 2618 is planning. It's planning a crossover design. It's planning a blinded design. The participating studies is called the Invictus study. This study from Decipher. In North America, it will be US and Canada. In Europe, these are the countries: Belgium, Finland, France, Germany, Italy, Netherlands, Poland, Spain, UK, Australia, and Singapore to gain additional geographic experience. There's also some discussion about, which they told me I could share with you today. Actually, I learned and then I'll share with you, which is that there's some discussion that the Decipher team is thinking about looking at DCC 2618 in a direct second-line study against sunitinib. Although that's a concept in development, they're calling it intrigue. So in summary, the new KIT and TGF receptor alpha-directed therapies, which have demonstrated promising activity in advanced GIST, particularly in D842V, as well as KIT mutant tumors. And their international pivotal phase 3 trials hold promise to further develop these opportunities into potentially new drugs in GIST. But again, I think that this is really exciting data. It's been a long time in GIST that we've had new drugs that have come into development in this way. Bill's going to talk to you about some other drugs that are really also, I think, very exciting and coming right along. And I think for the community, our job and our goals will really be to see the more exposure we get with these drugs, what do the toxicity profiles look like, what are the efficacy, how well are they working as you test them in more patients, and then how ultimately will we fit them into our armamentarium for disease control.
We're going to hold off on questions until after Dr. Tapp has his presentation. One piece of information I'd like to share with the advocacy group is Dr. Trent was responding to me earlier on the D842B, and he said that he had a special appeal. He said, when you have a patient that is in a GIST center, many times the doctors will go ahead and direct people into these trials. But if people are at home and with their local oncologist, many local oncologists don't know that this mutation is very responsive in this trial. So he was making an appeal that as advocacy groups, we try to direct people from the local oncologist to the trial center so that they can get involved.
Yeah, no,
so you probably have that too.
Yeah, no, but I agree. I mean, especially for the D842V patients, because the options, there really are no beneficial options for them outside of the trial up until this point. So I think for K42B patients, there's really a great opportunity here to have exposure to an effective therapy.